So now I want to talk to you about the first step in making a protein, and it's called transcription. Transcription happens inside of the nucleus. During this step, DNA is copied to create mRNA, and then that mRNA will leave the nucleus. So why does this happen? Why do we need this step? Well, remember that on DNA, we have genes, and those genes are the instructions for making a protein. Proteins are made outside of the nucleus. DNA cannot leave the nucleus. DNA has to stay inside, but it has to get those instructions to the ribosome, which is outside. Okay? If DNA can't leave and can't bring the message, then it has to find something else that can bring the message. And it uses RNA, specifically mRNA or messenger RNA. It acts as a messenger, and it's going to carry that message to where the protein is made. Just a couple of reminders about RNA, right? RNA is single-stranded, and RNA contains uracil instead of thymine. So when you're looking at the base pairing, if you look at that kind of orange colored square, if there's a G in DNA, its pair in RNA is still C. If there's a C in DNA, the RNA pairing is still G, right? So C's and G's are the same. If you have a T on the DNA, it pairs with an A. So that one is also the same. Here's where the difference is. If your DNA contains an A, the RNA base pair would be U, uracil. And you can see that in the image. Here we have an A, which pairs up with a U. Now, if you look at messenger RNA, okay, we already know that it carries the message from DNA outside of the nucleus, and it's going to bring it to the ribosome. Okay. Now, if you look at that message, we can break it down into groups of 3 letters. Okay, and we call those groups codons. Okay, so GCU would be a codon. ACG is a codon. Okay, and with those 4 letters, we can make 64 possible different combinations of those 4 letters. Okay, remember, so a codon is a group of 3 letters, okay, and there are 64 possible different codons. Now, there's this really handy codon wheel. The codon wheel tells you which amino acid matches up with which codon. So if you look here, here's our strand of mRNA. It's the message. This message is going to tell the ribosome which order it's going to assemble the amino acids. And so it reads the codons to figure out which amino acid goes where in the protein. So if you read the first codon, it says GCU. Okay, so what you do is you go to the codon wheel and you start in the center. And first you find the first letter, G. So I'm going to move over to G. The next letter is C. So I go over to C right here, followed by U. And then right at the end, it's going to bring me to the name of the amino acid that pairs up with that codon. So alanine. Okay, so in this protein, the first amino acid would have to be alanine. Okay, then what would the next amino acid be? You read ACG. So go back to the codon wheel, A, C, G. The second amino acid would be threonine. Okay, let's go to 3. Codon 3 would be G, A, G. Okay, G, A, G, glutamic acid. Number 4 is CUU, C-U-U, leucine. Okay, number 5, CGG. CGG, arginine. AGC, AGC, serine. On the last one, UAG, UAG, stop. Okay, that's interesting, right? What does stop mean? Stop isn't an amino acid. What that stop codon tells the ribosome is that your protein is done being made. There's one other special codon I want to point out to you, and it's AUG, methionine. AUG is the start codon. It's going to tell the ribosome to start assembling the protein.